Very good morning to you, Sir Fuad, um, and my fellow friends. So, I'm from Faculty of Health Science, Department of Environmental Health, Bachelor of Environmental Health and Safety once. So, my name is Fendi bin Mita, and my matrix is BHP 2404620 from cohort 23, and I'm currently conducting my training, practical training at Pejabat Kesihatan Kawasan Sandakan. So this presentation is about my practical training for which is food safety. So Food Safety and Quality Division or BKKM at Pejabat Kesihatan Kawasan Sandakan for the general objective is to ensure consumer get clean and safe food that is high quality, properly labeled, and protect public health. For the specific objective is to protect public against health hazard in food preparation and consumption, ensure food meets pollution and pre-standard with correct labeling, verify clean processing, storage, and also handling, regulate imported and exported food compliance, and also provide correct health information to users. So this is the related law used in the units. So the first one is Food Act 1983, Act 281. So this established legal framework for food preparation, sales, and distribution, ensuring all food products are safe for consumption. So the second one is Food Hygiene Regulation 2009. This specifies hygiene requirements, mandatory registration, regular inspection, and penalties for non-compliance. We will also have a Local Government Act 1976 and Trade Description Act 2011, which ensure accurate labeling and marketing of food products, prohibiting false claims about origin and halal certification. Okay, so this is the role of AEHO and EHO in Food Safety and Quality Division, BKKM at Pejabat Kesehatan Sandakan. So the first one is determine the preparation, sale, storage of all the foodstuffs is safe and also clean. Determining that all food items sold are free from contaminations or addition of unnecessary food additive and fraud, comply with the regulation and law set, label and advertise in a way is not misleading. And the third one is determine all the foodstuff imported into the country are safe and comply with the Food Act. And the fourth one is determine that food consignment exported uh, by this country comply with the law required by the importing countries. So the fifth one is determining that the public receives sufficient information on aspect of food safety. So this is my logbook activities 4.3 microbiological sampling. So my activity is to take food sampling for analysis in accordance with the Food Act 1983. So my target is ready to eat food. So microbiological sampling is testing food samples for harmful bacteria, virus, and other microorganisms to ensure food is safe for consumption and meets regulatory standards. Next, this is the checklist item needed for RTA food sampling. So make sure we have all this necessary equipment before conducting ready-to-eat food sampling for microbiological and chemical analysis. So first we must have a sterile sampling containers. Usually we use well pack, sterile swabs, sterile utensils for antiseptic handling, gloves for preventing cross contamination, alcohol wipe disinfectant, sampling form, cooler box with ice pack. This is to maintain the temperature during transport. Label and waterproof marker pen for sample identification, and also probe thermometer to record food temperature, or we can have a bottle of water as our reference bottle. Okay, and then the last one is we must have camera lah to for documentation. So this is the activity that we do, microbiological sampling activities. So this is actually a monthly surveillance sampling for meat for microbiological testing, which is conducted at the Orites restaurant and restaurant Seri Petani. So this activity ensure that food preparation area and ready to eat food meet safety standards and are free from harmful microorganisms. So this is the sampling location and also the procedure. First, we go to Orites restaurant Batu Empat around 10 a.m. to take sample daging masak kari. And then for the second sample, we take meat sample for daging masak rendang at restaurant Seri Petani around 11:15 a.m. And then we do documentation and laboratory examination after that around 1:20 p.m. at the MKMM laboratory Sandakan. Okay. This is sampling overview to see the purpose, test conducted, and also the significance. So the purpose is to assess the microbiological safety of the daging rendang and also masak kari served at the selected food premises in Sandakan. So for the test conduct, we do E. coli and TPC, total plate count. The significance is both tests serve as an important indicator of food hygiene and safety standard at the premises. So all the sampling activities were conducted by an Assistant Environmental Health Officer (PPKP), trained and authorized under the Food Act 1983. So before sampling, we first must see the owner, show our authorization card, and explain our objective is to take microbiological sampling at the place lah. And then after that, prepare and we must use all the protective personal equipment, which is PPE such as apron, such as head cover, and also glove. For the sampling methodology preparation, we ask for their ladle and sterilize it to prevent cross contamination, and then we do collection, which is the samples collected in sterile containers. uh, the sample containers that we use is well packed to following proper food safety protocol, and also after that we seal the well pack and we label. So each sample properly labeled with identification details, date and time and location. After that, we put the sample inside the cool box. We fill with ice to maintain appropriate temperature during transport. So this is the documentation process. This is the required documentation, which is request form for food sample analysis, form A, and food sampling notice, uh, sample labels with complete identification detail, also chain of custody documentation. So all the documentation was complete at the office following sampling activities and verified for accuracy before laboratory submissions. So as you can see at my slide, this is the proper documentation ensure traceability and legal compliance throughout the sampling process. So this is the sampling, delivery, and submission. We need to have a temperature control. The samples are transported in insulated containers with ice pack to maintain appropriate temperature, which is 0 to 4 degrees Celsius, to prevent microbiological growth during transit. And then laboratory submission. The samples delivered to MKMM Laboratory Makmal Keselamatan and Quality Makanan. And then digital recording sample information entered into Food Safety Information System of Malaysia, or known as POSIM, for digital tracking and record keeping. This is the result of the sample taken. So for analysis, result and interpretation, no E. coli detected in any samples, confirming adequate personal hygiene practice among food handlers. No total coliform detected also, indicating the preparation area were clean, the utensils were okay, and the water source is okay. So we also have total pet count. So that is within permissible limits under Food Regulation 1985, reflecting satisfactory hygiene practice. So this negative result shows satisfactory hygiene practice during food preparation, storage, and handling at both inspected food premise. So for the follow-up and recommendation, so we need to do official compliance notification. So we need to formally inform both restaurant of their compliance status, which acknowledgement for maintaining safe microbiological standard in ready-to-eat food. We also continue to routine monitoring, so maintain both premise in the surveillance cycle with periodic sampling of other high-risk food to ensure broader microbiological coverage. So the third one is strengthen preventive measure, encourage continuation of GHP and HACCP protocol with refresher hygiene training for food handler. So for conclusion, the negative result for E. coli, coliform and TPC confirm that the daging rendang and daging masakari served at both restaurant Orites and restaurant Seri Petani meets microbiological safety standard and prescribed under Malaysian food safety regulation. So this finding validate the effectiveness of current hygiene and food handling practices at the premises. So continued monitoring coupled with preventive reinforcement is recommended to sustain this level of compliance and protect public health. So that's all for me. Thank you.